Okay, so this is Brett Smith. Brett Smith comes from, what is it, Roanoke?
Round Roanoke.
Okay, Roanoke. And Brett may look young, but he's actually been involved in fungi a lot longer than several of us, probably many of us in here. And if you look at some of those photos, which we'll talk about those in a little bit, you'll see little young Brett in some of those photos. Did you see your picture in there?
I did not, no.
With John and Skip. So yeah, Brett's been around doing this for quite a while, and he was our keynote speaker last year for those of you who are kind of new to this event. And so this is our gift for Friday night is Brett teaching us about carnivorous fungi. So everybody shut up, listen to Brett, and he's going to blow your mind.
Thank you so much.
All right.
So I'm very excited to introduce to you your newest phobia, carnivorous fungi. Now, you've probably heard of carnivorous plants before, you know, the pitcher plant and Venus flytrap, but the carnivorous fungi get no media, and they're, in my opinion, about twice as terrifying. Before we get into that, I'd like to talk a little bit about some of the animals that I'm going to be mentioning in this, because some of them are a little bit obscure, and also just some general fungal anatomy, just to catch everything up. So this right here is a nematode. Most of the fungi that I'm going to be talking about in this presentation eat nematodes. There are these group of worms that are loosely related to arthropods, so shelled bugs, millipedes, etc. And they are usually microscopic, so they're not even visible to the naked eye. But this one happens to be macroscopic. And this over here is your Pleurotus boistris, preparing for a delicious meal over here. So, in terms of the fungal anatomy side of things, you have the mushroom, right? And this is what we generally think of. This is really just the reproductive organ of the fungus. Beneath it, in this wood, you're going to have hyphal strands all throughout. So it's like this massive root network, but it's so much more than that. Like if you take a tree, for example, this mushroom would be like the apple of the tree, and the tree itself would be everything underneath in the wood. And this came from along right back out there. This is some sort of oyster mushroom or Pleurotus species. I'm not totally sure which, to be honest. So starting off with some ground rules for what I'm considering a carnivorous fungi, because there are quite a lot of fungi that are entomopathogens, meaning that they are pathogenic to bugs. Some of the more common ones are cordyceps. So those are the ones that will pop out of the head of a bug. And this presentation would be pretty long if I included all of those. So this is just going to really talk about the fungi that are going to have their main fungal component. They're not going to grow inside of stuff for most of their life cycle. They're going to be pretty independent, and they're going to kill stuff on the side and consume those organisms as opposed to living in the insect as a cordyceps does. So right here, we have 3 of the common carnivorous fungi that you may have seen before. We have the oyster mushrooms here, we have the shaggy veins, and the wine caps, and you've probably eaten some of these. So, first one I would like to talk about is the oyster mushroom, this one right here. These guys are really interesting fungi, got a nice fruity smell. I love to see them, they're quite a treat. And they grow on dead wood, which means that they're saprotrophic or saprophytic, which means that they degrade dead wood and other organic detritus. In other words, they're mulch munchers. And the wood that they grow on does not have a lot of nitrogen in it. And proteins, DNA, pretty much everything, every complex molecule in your body is going to some way incorporate quite a good bit of nitrogen. It's one of the more abundant elements in your body. And it's the same thing for oyster mushrooms and all fungi, in fact. So they need some way to get nitrogen, and the wood's just not cutting it. So they've evolved this ability to prey on nematodes, you know, the little worm things that are quite abundant in the wood. In fact, I imagine that I'm holding quite a lot just in this small sliver of wood. They are on every continent, including Antarctica, in the driest places to the lowest depths. They're truly everywhere, and there's estimated to be around 1 million different species, where we only know around 30,000. So they are everywhere. They're super abundant, which makes them a perfect food source for this fungi. And they also happen to be packed full of nitrogen. So these fungi, they produce hyphae beneath the surface of the wood. I don't know if you can see any here. Probably not. Sometimes you can see a little bit of mycelium, but I cannot really see anything on this one. And what they have on these hyphae, these little thread-like structures, are these little, and the literature that I read compared them to lollipop-like structures that would grow on the hyphae, and they're known as toxocysts. And it's about what it sounds like. It's analogous to a water balloon filled with poison. So whenever you have a nematode, and they kind of crawl around erratically like this, they will be crawling through the wood, and they'll bump into one of these toxocysts. And this will burst. This little bulbous part will burst, and it will release a compound known as 3-octanone. This is a decently common compound in a lot of living things, but the oyster mushrooms use it in a new way, because this concentration of 3-octanone can apparently cause seizures and muscle spasms in nematodes to the point where they're no longer able to function. So they're either paralyzed or they're dead. And what the oyster mushroom does is it, once it detects that a nematode has bumped into one of these toxocysts, is it'll grow its hyphae out and into the nematode's body wall, which is quite terrifying. So it'll kill this prey, so it's like a bit of an ambush hunter, but not really, with these little traps, and then grow into it and then suck the nematode juices, so to speak, from the inside.
Can we eat the mushroom?
Yeah, so if you eat a Pleurotus mushroom, it's probably going to have just a little bit of nematode DNA in it, but, you know, extra protein. But this happens for almost all, and I say almost because there could be some undiscovered species or just understudied ones in the genus Pleurotus, so pretty much all oyster mushrooms in this genus do this. And then there's some other groups that are related to oyster mushrooms that do a similar thing as well. So I kind of touched on this just a minute ago, but just to recap, they do this whole thing. They've evolved this really complex and weird mechanism of killing nematodes just so that they can get a little bit more nitrogen, just so that they can have that little bit of edge. And it's quite similar to the way that carnivorous plants, or I should say the reason that carnivorous plants are carnivorous. A lot of them grow in bogs, and a lot of the nitrogen will either get seeped out or consumed or otherwise, and they just do not have enough nitrogen where they're growing. So they kill insects that are buzzing around and digest them for their nitrogen, not for energy, but for their nitrogen. These fungi do a lot of the same thing, at least these ones. There's a couple weirdos that I'm going to touch on near the end of the presentation. So another semi-common one that is actually carnivorous is the Stropharia. These are the wine caps, and this is pretty recent that they've discovered this. So they're mycorrhizal fungi. They're quite beautiful. They've got these beautiful yellow stalks, these dark-ish gills. They've got the ring around the site, the partial veil, and they've got a beautiful red cap. So they're a pretty choice edible because they're not super difficult to identify, and they apparently taste really good. In fact, I believe I saw its texture compared to potato or something like that whenever I was doing research for this. But what's not known about these fungi by most is that they are little killing machines. So beneath the surface, yeah, and the way that they do it is quite awful. They have these like mycelial networks, and they have the hyphae branching off of it. And these hyphae have these little spiky balls. So I like to think of them as like those naval mines that you always see in movies. Whenever you like dip the camera beneath the surface and they're all there, it's kind of like that. But imagine that beneath the ground and with a thread network of fungi. They have all these little spiky balls, or as they're called, acanthocytes. And they were originally believed to just be there for defense. So if something was going to try to eat the mushroom, it was obviously not going to have a good time doing so, at least subterraneally, not necessarily the mushroom bit, but the hyphae and the mycelium. So we originally thought it was to ward off predators, but as you'll see in this next slide, that's probably not what's happening. So this is a bit of a mess here, but what's essentially happening is that you have this little acanthocyte, these spiky balls, and a nematode kind of, I'm going to call it, swims into it, and the nematode gets punctured. And here's a little acanthocyte that's kind of detached, embedded in a nematode. Well, that's really all that needs to happen, because what happens is that the nematode and all of their guts leak out. I don't know how better to describe it, but it'd be like if you took a grape in your hand, poked a hole in the top, and then just squeezed it. Everything's going to come out, and all this like little kind of gray cloud here, that is nematode juice, which is a word that I'm going to be saying a lot in this presentation that I would have never thought that I would before. And then I believe that these are 2 puncture wounds. So they do this, and then the hyphae, the red parts, actually grow into the nematode, much like the oyster mushroom did, and consume it, or I should say slurp it, because this looks more like a smoothie to me than food. And then we have the fungi that decided that it would go overkill and then employ both of these methods at once. So these are the shaggy mane fungi. I used to see them in my backyard a lot, and I got very, very excited whenever I saw them because they're beautiful fungi, and they pretty much melt from the cap down if you're unfamiliar with them. It's really a spectacle to see. And a lot of people also like to eat these because they're somewhat easy to identify, because taxonomists can no longer decide on what's what with Caprinus and what is a Coprinellus and all that good stuff. But the shaggy mane has recently been discovered to be carnivorous. And the way that they do this, on the more mechanical damage side of things, are these little structures that are kind of similar to the acanthocytes. But I can imagine the conversation in my head where the scientists doing this research project named them quote-unquote spiny balls. That's their scientific name. They could not come up with a cool, fancy one. And in addition to that, they also kind of, in my opinion, have the Pleurotus beat, because they produce not 1, not 5, but 7 toxins that are all capable of killing nematodes, 2 of which we have not found anywhere else. So they just have 2 completely novel toxins just for this purpose, as well as these mechanical devices for puncturing nematodes. And then, much like the other ones, once the nematode is dead or wish it was dead, the fungi takes its hyphae and grows into it and slurps out all the nematode juice. I've been carrying this around the entire presentation. So, these are the 7 different, okay, I need to make sure that I did not miscount there, the 7 different compounds that Caprinus comatus, or the shaggy mane, produces to kill nematodes. I'm not honestly 100% sure which of the 2 are novel, but they're all apparently deadly to nematodes, and they're all quite interesting. Now, here are some of the more oddball fungi. These are just kind of touching on it. This is a family, I believe, called the Orbalaceae, and here's them growing on a dead log. This specific species is Orbilia xanthostyma, and there's not really much information online as to whether it is carnivorous or not. I just could not find any studies referencing it as a carnivorous fungi, nor could I find any counter evidence. But it is one of the ones that produces these bright fruiting bodies that would look pretty on a slide, essentially. So these are sac fungi, the ascomycetes. These are closer related to morels than the shaggy manes and wine caps and Pleurotus that we were just talking about a minute ago. These are closely related to your morels, your cup fungi, and etc. But within this family, there are quite a few, actually quite an alarming amount of genuses that are capable of killing nematodes in many unique and horrifying ways. And they utilize these nematodes for nitrogen, just as the other ones did. So it's kind of the same purpose and somewhat similar mechanisms, but in a completely different group. And we call that convergent evolution, where 2 completely different things evolve to have similar traits because they're living under similar environmental stresses, particularly not enough nitrogen in this case. So they both thought that the best way to go about it, well, I shouldn't say thought because evolution does not think, but the way that they went about it was learning how to lasso nematodes 90 50s cowboy movie style. So this is the Arthropoda. This is a particular genus, and I believe there's one other in this group that does this. These are the nematode lassoing fungi, as I like to call it, but it's really more analogous to a life preserver. So these do not produce vibrant or visible fruiting bodies that I know of or that I could find on the internet. But what they do produce is these horrifying devices right here, what were called constricting rings for a good reason. So they're made up of 3 cells. So you can think of this as like a life preserver. Each section, you have one section, you have another, and then you have a third one. What happens is a nematode will accidentally swim through that, and then the life preserver, each of these 3 cells, inflates to about 3 times their size, which essentially constricts the nematode and holds it in place. Now, I'm not sure if this does anything in terms of literally choking the nematode or preventing the movement of important chemicals from one place to another in its body, but what it does do is it holds it in place long enough for the fungi to take its little devious hyphae and puncture its body wall and drink the nematode like a juice box. There's a couple other things in that genus that also, before I move on, create club-like appendages. These are the only ones that have what are called active mechanisms or active trapping mechanisms, which means that they, whenever a nematode essentially toggles the trap does something. The other ones in this group, a lot of the other ones in this group, just have what are called passive traps. So that'd be more like a tripwire as opposed to a laser-guided gun in a corner or something along those lines. But essentially what's happening here is, or in these other groups, is that you have these little clubs that stick up out of the hyphae, and a nematode will run into it, and the nematode will get stuck on the club, and then from there, the hyphae pretty much eat it alive. It grows into the nematode. And other ones have these vast webs of just this sticky snare-like trap, kind of much like a spider web. So whenever the nematode runs into that, it gets both entangled and ensnared, and then the fungi is able to digest the nematode. Now, fortunately for the nematodes, they're not the only one on the menu. Here we have something that makes me very sad because I love rotifers. They're another really neat group of animals, and they're called wheel animals. Most of them are microscopic. I believe that there are a few that if you're really squinting, you can see with the naked eye. And then there's an entire group that is like parasitic of large animals that get like a foot long or something, but I digress. So you have these rotifers, and they are usually a little stalk-like shape with like a flower almost head, and they have cilia. So they're hairs that can beat back and forth like this, and it brings stuff from the surrounding soil, if it's waterlogged enough or whatever substrate they're in, usually water, into their mouth, and then they can digest it. And then they have, at least the deloid rotifers, have this little base at the back where they can attach to stuff. And if you watch one move, it almost looks like they're slinking along. They're really neat. But there's a couple of fungi that can actually consume these rotifers, and they've developed quite the method of doing so. It does not seem like much until I read a couple slides ahead, so bear with me. What we have here, this long, strand, if you can see that, is Zoophagus. This is a genus of fungi of somewhat uncertain placement. I saw some places put it in Ascomycota, others in Zoopagomycota, which is a group of molds, and then yet others just say that they did not know. So I put it in Zoopagomycota, or Zoopagomycetes, because that seemed like it was the most common classification that they put this fungi into, but it could be an Ascomycota, so just bear that in mind. There are these long strands right here, and then you can see these little protrusions coming off to the sides. Ignore this little poor rotifer right here. You can see these little protrusions coming out to the side. These are little club traps, and it would almost be like if you took a baseball bat and then put a bunch of fly paper and then surrounded it in that. They're really sticky and adhesive, and the rotifers get their hairs, these cilia, stuck on these club shapes, because you can imagine their cilia are going to very easily stick to something this adhesive. So it's going to stick to it. And then what happens is that the wall between the club and the hyphae itself kind of breaks down, and it stops being an external structure and literally becomes part of the hyphae. From there, it turns into what's called a haustoria, which is essentially a fancy name for a little tube that some things will use to pierce their hosts or prey to suck a bunch of juices out of them. So these form into haustoria and then become part of the hyphae itself and grow into the rotifer here. But what's interesting is some species reactions, and there are supposedly about 4 different things, well, 3 different living organisms and one that's debatable, that interact in this relationship here. So there's obviously the rotifer, which is having quite a bad day right here, and the fungi, which has little clubs sticking up. So the rotifer gets caught on the hair. But what the fungi has is a bunch of these little bacteria, and I believe that the genus Bacillus was named in a couple studies, or in one of the main studies that I was reading. These bacillus that will get injected through the haustoria, through the little club-like protrusion once it enters the rotifer, that are really good at degrading it. But that's only 3 species. The 4th one is a virus. Well, it's kind of a virus. It's more already incorporated itself into a bacteria. So there's this group of viruses called the bacteriophages. They look like something out of a 1990s sci-fi horror film. They're this little, I think it's called an isodecahedron or something on top. So think like a soccer ball, but more polygonal. And then you have a long thread looped around a pole coming down, and then 8 spider-like legs coming out. If you've ever watched the first Despicable Me movie, it's like those little cookie-cutter machines that Gru releases to go cut a hole in the wall. They look exactly like that. And what they'll do is they'll inject their DNA into bacteria. Sometimes they'll kill the bacteria, and other times they'll incorporate their DNA into the bacteria's DNA. And if you think about this is like winning the lottery for the virus. The bacteria reproduces, and so the virus that's incorporated itself into the bacteria's DNA gets a free ride. It gets to reproduce with the bacteria. But sometimes these prophage genes are special, or they're called prophage genes, these genes that got injected by viruses. Quite a couple of them are the reasons that a lot of common diseases, like E. coli, are infectious. So these prophage genes actually allow them to infect us, and in this case to produce venom and toxins that are able to degrade the rotifer. So there was a large amount of these bacillus that they found in these little clubs that had these special prophage genes that allowed them to produce a lot of chitinase, which is an enzyme capable of breaking down chitin, as well as quite a couple other number of different things that came from a virus gene that got injected into the bacteria. And they found more of these bacteria that had these genes inside rather than outside of the fungus, so it must be something significant. So they inject these bacteria that have these virus genes that are able to make toxins into this rotifer to break down the rotifer so that the bacteria and fungi can both eat. It is an insane example of symbiosis and, I suppose, carnivory as well. It's just a ridiculous way that some different creatures in their environment interact, and I think it's really impressive and quite terrifying at the same time. So this one also makes me especially sad. I have a soft spot for these cute little guys right here. They're known as the springtails or the columbola. I believe that's how you pronounce that. And it is a group that's adjacent to insects. They're not technically insects, but they're hexapods. They have 6 legs, and they're quite tiny. and they are all over the place. If you grab a bunch of leaves out there and then look at it really closely, I'd be willing to bet that you'd see at least one or two of these springtails wandering around. And they're really good at breaking down and consuming detritus, so they're detritivores, and they're tiny. So what this one fungi right here, Lacaria bicolor, does is it has these hyphae that are able to kill these springtails. We have no clue how. There is just a couple of studies reporting this, and it's relatively new. So some of this data is a little bit tentative, but we're fairly certain that this is a documentable thing that happens. Now, the Lacaria bicolor are symbionts with white pine trees. So their mycelium connect to the roots of the pine trees, and they exchange nutrients back and forth. And so, is there a slide on? No. What these fungi do is they'll kill these springtails, and they'll absorb their nitrogen, as most of these carnivorous fungi do, and they will pass a lot of that nitrogen onto the tree. So they're not actually keeping these nitrogens, this nitrogen. They're being a bit of a thief from the springtail, and then they're giving all this nitrogen back to the tree. So they tracked this nitrogen that was in these springtails, and they found that, I believe it was around 25% of the nitrogen that the white pines absorbed was from springtails, which is more than you normally expect. Because springtails are going to die, and then the tree's going to absorb the nitrogen. But what they found was whenever they were with these fungi here, they would actually absorb more nitrogen from springtails than otherwise. So these fungi, as they've observed killing springtails, so they've put populations of springtails in with the fungi, and it did not end well. They've observed the fungi killing a springtail, and they've also observed a lot of going onto the tree. It is another really crazy example of symbiosis. So this is one of our last groups here, the Lecophagus. Now I have this titled "Some Fungi Are Not As Picky," because these guys can eat a wide variety of stuff. You can find these fungi in many, many biomes. So this one group, Lecophagus, I believe, Arcticus, or Antarcticus, or some relative, was found eating tardigrades, so the little water bears or moss piglets in Antarctic lakes, whereas others you can just find in the holes of trees that capture water. And they usually use this similar club mechanism, so the tardigrade or the rotifer will get trapped on them, and they can also, of course, digest nematodes, because most of these fungi do. Yes?
No.
Lecophagus antarcticus. Oh, do you hear that?
No, I was just sad that they ate the tardigrades.
Oh, there's an owl. I'm sorry.
Oh,
just didn't want to.
The screech owl?
I apologize. I just thought that you all would want to hear that. So these guys are especially devious in the way that they go about it, because they have these clubs that release this sticky substance that the tardigrades think they can eat. So the tardigrades think that this fungus is providing them with a yummy snack. What happens is that the tardigrade, with this cute little mouth right here, this makes me really sad, tries to bite down on the clubs that this fungi produces with its hyphae, and the fungi then proceeds to inject it, grow into it with haustoria, and then assimilate, as I have written down here, the tardigrade into the fungi, which is a fancy word for sucking all the tardigrade juices out of the tardigrade. And that is the formal end of the presentation. Here are some of the citations that I used here. Do we have any questions or anything that we would like to discuss? Sure.
So with the Lacaria, are they eating like adult springtails or like in their larval form, or does anyone know that yet?
I'm not personally 100% sure. The study could have mentioned it and I just kind of glazed over it. but I would imagine you're right. They'd be more likely to eat younger springtails.
Or
That's
in
a
the soil.
yeah. That's a good point. They'd probably eat either species that live most of their life like in the soil. Like I guess they can live in more loose soil, or their larvae. Yes, sir.
Are any of the toxins that get produced something that can affect humans, or is it on such a microscopic level because it's directed at springtails that it just is a dose thing where it's not enough to affect humans? In any of your reading, were any of those bad news for us?
So they did.
Doses?
Oh, sorry. They did not mention it. But of course, it's kind of the toxin makes the dose. Like the amount that Pleurotus produce of like 3-octanone would not be even close to enough to kill a human. Hence, why we eat them, and probably ingest some of their hyphae and are totally fine. But I'm sure that if you took a gallon of distilled 3-octanone, you're not going to be having a good time. But I'm not totally sure if they're especially poisonous to humans. But I believe 3-octanone is somewhat commonly found in nature. So I'd say that some of these are quite specialized, especially with the Caprinus and some other ones.
Sorry.
no,
So
you're
with
fine.
the Pleurotus, the 3-octanone, you said your slide said it causes a calcium surge in the nematodes?
Yeah.
Is it essentially calcifying the nematodes?
Not really. What I meant, it's in the muscles, so it's causing the muscle channel, like the the ion channels in the muscles to take in a ton of calcium, and then that causes the muscles to tense or contract, which can paralyze the nematode, kill the nematode, and etc.
And how fast on the scale is this happening from it, like, capturing the nematode to, like, digesting it? Does anyone know that?
Fungi don't move real fast, but the nematode is killed in pretty close proximity to the fungi. So I imagine it's not going to be like in a matter of like seconds by any means, but I'd imagine like somewhere on like the scale of like an hour at most, because these can explode about instantly. The nematode dies, I believe it said in like 5 or so minutes. At that I'm thinking about a different statistic. And then it's just a matter of the fungi noticing the nematode and then growing hyphae towards it. And I use noticing very, very particularly because my other talk idea was, are fungi conscious?
Next time.
Next time. Come back for more.
The 3 ring cells?
Huh.
The 3 ring cells?
Oh,
of
yes.
Other species? You said that they inflate, but they don't. It must be some kind of liquid. What is moving?
So, I imagine the way that they're drawn, so you can see that they're kind of attached to like a hyphal stalk. I imagine that there is like decent cytoplasmic flow between the hyphae itself and the ring, the constricting ring. So I imagine that the fungus could pump a bunch of cytoplasm or some sort of fluid into it. That being said, it can happen on the scale of 1 to 2 seconds on the ring contracting. So it's very fast in terms of fungi, right? But that's a good question, but I'd imagine that it's something to do with this little stalk connecting here and some sort of cytoplasmic flow.
They're
I'm sorry.
also like aquaporins, in
Yeah.
cells, so maybe it opens up water channels and just puts a bunch of water in them.
Yeah, that could make happen. That could happen, especially if there was some sort of, like, if these were, like, almost, like, in a deflated state, like, I imagine that it could inflate really fast, or it has some form of, like, more active transport. But yeah, aquaporins could potentially do something like that. I'm sorry, what were you going to ask?
The rotifers, when you were talking about the rotifers, is that happening in water?
Oftentimes, yes. So the Lecophagus They like to live in really, really moist environments, as well as lakes and ponds and like just straight up water. So yes.
It says Shaggy's mane produces 7 compounds. Are there like primary and secondary compounds, or do they all work equally?
So I don't know how effective they are compared to each other, but these were the compounds that they used to neutralize the nematodes. So all of these play some role. I don't know if they work together. I don't know if the scientists know, but they're all toxic to the nematodes. But I don't believe that any of these are just kind of like byproducts, if that's what you're asking.
Yeah. Okay.
Yes, sir.
Do the mushrooms that kill nematodes kill certain species of nematodes, or are they kind of generalized?
So I did read something about some of the ascomycete fungi that are carnivorous doing something like that, like preying on particular species, but they did that in kind of a lab setting. So I imagine they're not super picky. They did observe some nematodes actually being able to resist the Pleurotus's toxin, I believe, and are able to consume it, either that or one of the other toxin-producing ones. So there are certainly some nematodes that are able to turn the table on the mushroom, but I do not believe so. Yes, sir.
You just mentioned that some nematodes can resist the toxin. Is that some species of nematodes or some individuals? Because, yeah.
Some species.
Okay.
I imagine that there would be some individuals that are more, able to withstand the toxin than others. But yes, I believe it's like distinct species and even genre.
Have you ever tried to notice these things on the microscope?
I am not good with fungi microscopy, to be honest. I don't know how you would be able to do that just because of my own lack of knowledge, but I have not. You could probably ask someone like Shannon Nix, and she'd probably know something about that because she does a lot of microscopy, but I have not personally.
Yeah, I would love to see that.
Oh, I know.
I mean, that would be so cool.
I mean, I'd feel bad for the nematode the whole time, but it would be interesting to see.
Yeah.
I don't know if there are like any videos on
There
YouTube.
are videos of this.
Yeah.
Really?
Oh,
Okay.
they're so cool.
Oh, okay.
Especially
Okay.
the lasso.
Yeah,
Goes in there and grabs them.
pretty much.
Wow. Any other questions?
I have a question.
Sure.
Fungus that isn't carnivorous presumably requires nitrogen as well. Do you know
Yes.
where those species can get different nitrogen?
So most of the non-carnivorous fungi are going to get it from their substrate. So the mycorrhizal fungi are going to get it like directly from the soil. Like temperate forests, which is pretty much what we're in at the moment, generally have a good bit of nutrients in their soils. So they're able to absorb that nitrogen from their surroundings. It's just more the ones in areas where they cannot absorb nitrogen that they have to resort to carnivory.
The one that shares nitrogen with the white pine trees, will it do that like on an as-needed basis, or is that a constant?
I would have to imagine it'd be on an as-needed basis, because if you had just constant nitrogen coming in, that's not going to be good for you.
Yeah.
You can have too much of a good thing, I suppose. So I'd say yes. Either that, or it's at a steady enough rate that it's not harmful to the tree.
Okay.
Like, that's just kind of an educated guess from my perspective. If there's no more questions, I believe that we can wrap up this presentation. Thank you all so much for coming tonight.